Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Eugenia, and I'll be your host. It's our pleasure to have LTS with us today, who will be presenting this webinar titled API Feasibility Studies for TTS and OTF. Today's guest speakers are Julia Lodder-Gadaczek, Director, Head of Laboratory, PHA Skin Lab Analytical Development R&D at LTS, and René Rietscher, Director, Head of Laboratory of Pharmaceutical Development R&D LTS. And now I'll provide some housekeeping information before we start. First of all, I would like to welcome you to our webinar platform On24. You will notice that this webinar is browser-based, so if you disconnect for any reason, please just click on the link that you received via email to rejoin the session. During today's event, there will be plenty of time to ask questions to our speakers. You can do that via the questions widget on the left-hand side of your screen. Just type your question in this box and click Submit. We will allocate some time at the end of the session to answer questions or any thoughts that you may have. You can download and review a PDF copy of today's presentation at the bottom of your screen. We would also like to invite you to fill out a short survey, which you can find in the survey widget. If you require any assistance, please use the yellow Help widget. And you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. And now, please allow me to welcome René. Over to you.
So thank you very much and welcome everyone to the LTS webinar on API feasibility today. And thank you for joining. A little bit on LTS as a company. LTS is the Transdermal market leader and leading global partner as a pharmaceutical industry in transdermal therapeutic system and oral thin films. LTS has over 1,600 employees and over 400 million euros of revenue last year, and out of this revenue, we reinvest over 30 million euros back into the organization to, for example, further develop our technologies. Overall, we have the capacity to produce up to 2.5 billion units per year. We have a global presence for our partners with 4 manufacturing sites with our headquarters in Andernach, Germany, which is roughly 1 hour from Frankfurt Airport. We do have 2 manufacturing sites located in the US at St. Paul and West Cortville, and a newly acquired site in Netanya, Israel. Furthermore, we have a small business unit in Shanghai for our business dealings in Asia. LTS is a CDMO and technology company and provides different dosage forms. For the webinar today, we are focusing on oral thin films, OTFs, which release their API via the mucosa of the mouth, and transdermal therapeutic systems, which are applied to the skin and act systemically or topically, which includes somehow also the topical patches. Furthermore, we are also developing micro-aerial patches, which have tiny needles that penetrate the dermal layer, dissolve, and release their API over time. Our newest technology is the Xarel wearable technology, which is used for home delivery of high volume and high viscosity formulations. So what could be the benefit of such feasibility studies of LTS for you? Usually pharmaceutical companies try to develop and administer their active substances via tablets or capsules. However, during the product development and also preclinical clinical studies, they are somehow faced with challenging properties like low oral bioavailability, adverse effects, limited drug exposure, or short half-life. Furthermore, suboptimal PK profiles during the preclinical and clinical studies could occur. In this case, an OTF or TTS feasibility study to investigate the potential of a transdermal or transmucosal candidate could be beneficial. The other use case could be the product life cycle management or marketed approved dosage form reformulated. We can furthermore improve certain aspects like the bioavailability, the drug delivery profiles, the compliance of the patient, or even reduce the dose frequency. I'm looking for example for transdermal systems. So let's start with our in-silico simulation tool, TRACE, assessment of a transdermal and transmucosal candidate. The basis of TRACE is a database. You can see roughly here, where we collect all available information like physicochemical properties, pharmacokinetic and dynamic parameter, and all further information like medical need, stability data, or further restrictions we find during our literature research or get from the certain company. Afterwards, we assess the information based on the available data and based on over 40 years of LTS experience. And furthermore, we have also an audit trail and approval system for transparency, so that all entries and trace analysis are at all steps traceable. So let me show you two case studies. First of all, for the trace analysis of azanapine to predict the transdermal permeability. Every aspect you can see here in this bullet point, like type of action or the indication, molecular weight log P, or even the advantage of such a system, so-called medical need, are visualized in the trace analysis here as spheres with individual weightings. And in summary, we collect all this spheres and weightings and bring them in a total trace score. Furthermore, you can find on the right side of the trace analysis further assumption and additional notes, like potential administration times of the patch, technical or stability restriction we found during our literature research. Please apologize that I cannot explain every aspect here in detail due to the limited time, but feel free to contact us so we can perform such a trace analysis also for your transdermal or transmucosal candidate. In summary, the available physicochemical properties of a candidate is most important for such trace analysis, which leads for azanapine to a positive in silico evaluation for transdermal drug delivery. The second case study you can see here was performed for esketamine hydrochloride to predict the oral mucosal permeability in this case. For oral mucosal evaluation, we consider slightly fewer evaluation points, and also the individual weightings you can see here adjusted as a barrier. The oral mucosa differs from the skin for transdermal application. The overall advantage for oral mucosa application is that we can use the common APIs API salts, in this case, the azanapine hydrochloride, as we need to use for transdermal application usually the free form of the API or the base form. So overall, we can say for the trace analysis of S-ketamine hydrochloride, this leads also to an positive in silico. I will now hand over the API to our in vitro feasibility lab. So Julia, it's your turn.
Thank you, René. Thank you. So after this perfect overview about our in-silico tool, we will come now to the practical approach of a feasibility study. And we want to see today tools and method for the API evaluation for potential transdermal system, and in the second part also for an ultra-thin film. Please keep in mind that we talk about a very early feasibility study, so we don't have the focus on prototype development in this part. We only focus on the permeation potential of an API directly, and so most of the experimental trials are based on API solutions in different ways. If we talk about the transdermal therapeutic system, then we normally have a patch which is applied to the skin. We have then the liberation of the drug and the absorption of the drug to the skin. So we have to overcome the stratum corneum as a barrier, and then we have the distribution in the epidermal layer, and then we have the uptake of the drug between the epidermal layer and the dermal layer, normally to the vesicular system, if we're talking about a transdermal system. In case of a topical system, we don't have any distribution to the vesicular system at all. So we have more a local distribution in the epidermal layers normally. As René mentioned before, for the application of transdermal therapeutic systems, the drugs need very special physicochemical properties. So molecule size cannot be so big. We need a special type of polarity and log-peak because we normally have a penetration route through the paracellular route along the alkyl change of lipophilic substances in the skin. You can see on the left side. And these are also controlled by tight junctions. This paracellular route is a main route for transdermal drugs. So over 90% of our drugs will follow this penetration route. The follicular route and also the transcellular route have only a minor role. Sometimes it can be if you have a liquid API, or we work with a nanoparticle, for example, that we have a mixture of follicular route and also of paracellular route. So if we want to mimic this uptake of a drug over the stratum corneum through the epidermal layer to the vesicular system, then we need also a good model. So normally we investigate this in vitro permeation studies, and we use the diffusion cell model as our principle. In a diffusion cell model, we have a donor compartment and an acceptor compartment. In the donor compartment, we normally apply the drug, or at the end, the formulation or patch. Then we have the penetration through a barrier. In this case, we use skin as barrier, and I will come to on the next slide to the models we can use. And then we have the transport to the acceptor compartment, where we mimic the vesicular system by using aqueous buffers to lose them for the systemic resorption. We quantify the API in the acceptor compartment at different sampling time points and get something like a penetration flux profile at the end to evaluate the potential of an API to be a permeation candidate for a transdermal drug, as a transdermal drug or not. you can see that we have the in vitro permeation studies, which has, let's say, a little bit time consuming. And so it's not really a good alternative in the very early feasibility study, where we have to prove different conditions for the APIs, where we have to screen many different enhancers to increase the permeability of an API. And so in this case, we normally start with the using of a micropermeation system, which is based on an artificial membrane. You can see it here on the right side. So we used Pampa. It's a commercial available system, short for parallel artificial membrane permeability assay. And what you have here is a micropermeation in a 96 well scale. And you have a membrane here, which functions as a barrier. And this membrane is based on different lipids, which are the main lipids in the stratum corneum. And so you have here a very good simulation of the stratum corneum at all. As I mentioned before, you have a micropermeation system, again, an acceptor compartment and a donor compartment. On the donor compartment, you can apply your test compound. Then you have the membrane as barrier, and in the acceptor compartment, you can quantify your API at different sampling time points. Another advantage of this model is that we not need so much time as we need for an in vitro permeation study, because normally if we work with an in vitro permeation study, then we would apply our API for the time of applying the patch, so 1 to 7 days normally. In this model, where we only have a membrane, we are finished after 6 hours. Another benefit is that we don't have any cross-contaminations with skin, so we don't need a full developed HPLC method to investigate here the quantification or the quantification of the API, and we can use a very fast photometric measurement, and we have a very fast result after 2 days. I mentioned before that in the in vitro permeation study, we use normally skin models, and we have two possibilities to use skin models as barrier function. So first of all, you can use human skin. In Europe, it's common to use human skin from plastic surgeries. I know that in the US, it's more common to use cadaver skin. This is the most predictive model for, let's say, in vivo conditions and in vivo studies, and we often say human skin is our gold standard for in vitro permeation trials. But you also have the disadvantage that you might have a high variability from donor to donor, because there is a difference from, caused by the gender, age, tissue, and dermatological quality of the skin. And so you have to set a reference at some time point, and you have to mix up your donors at the end. So it's good to know how is your value or ranking of permeability from donor to donor. So set a reference in the beginning is the best way to prove this. An alternative model is the use of Göttingen mini-pig. You have here a high comparability to human skin, and you can also see this in the structure on the bottom line. The benefit of this model is that all the animals are genetically similar. We only have one supplier and only one breeding conditions for every animal we get. And we also can choose always the same age and gender. So we don't have a very high variability from donor to donor, but at any time point, it's good to make a comparison to the human skin, because I said it in the beginning, sometimes you have APIs which also use a follicular route, and then you have an increase of permeability on the mini-pig because the hair follicle density is much higher than on human skin. I said before that we can use Pampa and or in vitro permeation studies also to select enhancers for your API. to be a potential candidate for transdermal therapeutic systems. And I will only want to clarify that we have to separate enhancers in different categories. So we have enhancers which has more a thermodynamic effect and maybe act as co-solvents for your API. And we have enhancers which has a function directly increasing the penetration in the skin, so classical penetration enhancers. And not every type of this enhancer can be investigated in the pamper, because if we have an enhancer which has a direct interaction in the lipid double layer in the skin, so for example, it is an enhancer which makes a lipid extraction or a fluidization of the skin, then we don't have the possibility to investigate this in pamper, because then we lost our membrane here. And so we have to go with a couple of enhancers directly to the in vitro permeation study to investigate them at all. After this short introduction about the tools we can use, I will come to the, let's say, overall procedure we normally apply. So if we start at any time point with week 1, where we have the theoretical evaluation, we have a closer look to the physical chemical properties of your API. We search for some HPLC methods which are available when we don't get them from our customers, and we can choose some enhancers based on the properties of the API we get. Then we can directly start with the development of an HPLC method. This is based on the, let's say, properties for our in vitro permeation studies. We also test different solubilities, receptor, enhancer, organic solvents, for example. And in parallel, because we don't need the HPLC method, we can also start with pamper as our screening model for pH dependencies, tolerabilities, receptor conditions, and also enhancers. And then normally we go more or less directly after 4 to 5 weeks to the first in vitro permeation studies. In the first in vitro permeation studies, we normally work with saturation concentrations, or we make a concentration series of your API. And at the end, when we have a good picture about potential enhancer we can use, we also have the possibility to combine different enhancers in our in vitro permeation studies in different ratios to see which might be the best result, or which might increase the permeability of your drug in the system. We can also investigate the residue contents, make full mass balance, and we can vary the application time, for example, if it's necessary. And after approximately 12 weeks, sometimes we are a little bit faster, depending on how much information we get in the beginning, for example. Then we have a full picture about your API, the potential of permeation, and we can make a go or no-go decision for next step. Next step means first prototypes and development and adhesion searching and so on. If we follow this procedure, then we normally start with the solubility of the API, and we need different solubilities at information. So we need the solubility in receptor medium, because we work here with an aqueous buffer system, and normally the substances are more lipophilic. So sometimes we have an issue with the solubilization of our API. And then we have the possibility to add some solubilizers also in the aqueous buffer system. But it's also necessary to know this for the in vivo conditions, because sometimes it can be that you have a very lipophilic substance which don't have the possibility to reach the synaptic vesicles or the bloodstream, because you have then a skin depot effect. And so we can choose this a little bit, or we can see this a little bit also in the solubility in aqueous systems. We normally investigate also the solubility in organic solvents. This derives a little bit to the decision for adhesive mass in the next step, for example. And we measure the solubility in different enhancers, which we can use as co-solvents, for example. Sometimes, you can see it here on the left side in the pictures, we also get a very first hint of first developability. In this example, we have a different pH aqueous buffer system, which we investigated for the solubility of this API, and the API itself is colorless, and we saw that there was a color change depending on the pH. So this might be a hint that not in every pH this API is stable anymore. This is an example, or a very easy example, of how a PAMPA experimental trial can look, or how is the profile of this PAMPA experimental trial. So we have the possibility to screen 32 samples in parallel, and we can select, for example, the best enhancers from the PAMPA experimental trials. So we select here, for example, the best 3 enhancers with the highest permeation profiles we get in this experimental trial. This example shows you that we have a very good discrimination in the PAMPA, which is comparable to what we expect in the in vitro permeation study on human skin. So it must be clear that the absolutely amounts in PAMPA and in human skin are not the same, a little bit dependent on the concentration we use of your API, and also that we only have a membrane on the one side and a full epidermal system on the other side, and this has influence on the absolute permeable amounts. But you can see that the ranking in both experimental trials is more or less the same. So the comparison of the PAMPA results to the in vitro permeation studies are normally direct, and so we can use the best combinations of enhancers which we evaluated with the PAMPA system also for first experimental trials for the in vitro permeation studies, and then go further on with the feasibility. If we are sure that we found a couple of good enhancers to increase, for example, your permeation, or we have the possibility that we have a very good co-solvent for your API, then it also makes sense to make some mixtures of your of the preferred enhancers. So in this case, you can see in red that we have here an enhancer with a very high solubility for the API, but no permeation potential or no increase of permeation potential for the API. And we have here in gray the enhancer with a permeation potential for the API, but with a low solubility. And if we combine this, also different ratios of both are possible, then we normally get something like you can see here in green. So we have a very high increase of the permeation profile because we mixed up a very good penetration enhancer with a very good co-solvent in this example. This is a second example for this. So we have here the opportunity that we have a very fast Cmax with the one enhancer and a very low profile with the other enhancer. Again, the low profile is with the enhancer with high solubility for the API, and the other one is the enhancer with low solubility for API. And you can see here after the Cmax, we don't have any more concentration pressure. And so we have a very, let's say, a direct, decrease of permeation. And if we combine this, it's not so clear to see, but I think it's clear, then we have the possibility to reach also a high Cmax, but we don't have this rapid decrease at the end. And so we can find out in the feasibility studies what are the all, and if we can mix up for some permeation or penetration potential at the end. Last example for the drug substance for transdermal therapeutic system. So in this example, we screened 11 enhancers with a solubility over 33% for the API. We only go with the 2 best enhancers. And because we only have the opportunity to go with 2 enhancers, we decided to go directly in a very simple prototype. So only with 1 adhesive, not at all more. And you could see again, without any enhancer, you have a permeation profile like this. And with the combination of both enhancers, you increases your permeation profile. To sum up this part, with the combination of the in-silico tool, the PAMPA, and the in vitro permeation studies, we have the possibility to find out in a very short time frame how is the potential of your API to be a candidate for a transdermal therapeutic system. And at the end, we have the possibility to make a go or no-go decision for the next step. This is not only based on this, let's say, result, it's also based on your target doses and, let's say, framework conditions you want to have, but we can get a very good picture about your potential, about the potential of your API to be a candidate for a transdermal therapeutic system. We will now come to the feasibility studies for orothin films. I think you will see that we have a lot of similar tools and methods for the feasibility, but we will also see that we have some differences, and I hope I can point out this difference at the end. So if we talk about an orothin film, then we normally talk about a drug substance, which will be carried over the transmucosal epithelium. So we have, again, a paracellular transport as our main route for this API, but it's necessary to know that we have a non-keratinized epithelium, so it might be a little bit easier to overcome the barrier, and we don't have a real lipid double layer structure. We have more a liposome vesicular system here. Orothin films can be applied at different sites of your oral cavity, and normally we're talking about a buccal or sublingual application. So structure of the non-keratinized epithelium in both sides is more or less the same, but we have, let's say, a different diffusion area. So in the buccal side, we have a lower diffusion area than in the sublingual side, and we also have here the possibility that the IPA can spread a little bit more if we apply something sublingual. This is necessary to know because we will later come to the models we can use. So if we want to model the transmucosal penetration, we need, again, an in vitro permeation model. And for this in vitro permeation model, we also need a barrier. In case of the oral cavity, we have not the possibility to work with human tissue. And so we investigated and established, I think a few years ago, to use animal model, and we normally use as our barrier the mucosal esophagus tissue from pigs. You can see in the pictures of the structure that we have more or less the same structures here. Sometimes it's necessary to investigate what happens in a living model, because we are talking, in case of skin and also in case of tissue from esophagus, that we have a non-living model at all. But maybe your API has a high potential, or we have an active transport or a metabolism directly in the non-keratinized epithelium, and then it makes sense to investigate this also in a living model with a full enzymatic activity. And we evaluated a lot of different models, all based on the same principle. They are all based on reconstituted non-keratinized oral epitheliums, but the one from MATEC is the only one which derives from primary cell cultures, and it has the best barrier function at all. We need barrier and we need an in vitro permeation model. So classically, we also have the possibility to use diffusion cells, but diffusion cells, in case of investigation, an API substance for orothin films has some disadvantages. Caused by the tissue we use, we have a very high risk of leakage when we apply it to a more normal diffusion cell. And what else you have to know is it's a sampling of a diffusion cell took longer time. And if we are talking about the application of a patch, then it's not a problem at all, because normally we will apply patches from 1 day to 7 days or many hours. But in case of an oral thin film and a very fast uptake of your API to the tissue and through the epithelium to the vesicular system, then we talk about seconds. And so we need a model which allows us to have very short sampling times. And so we adapted cell culture equipment for this. So normally we use a 6-well plate deriving from cell culture experiment. You can see it here. And we use cell culture inserts where we mount our mucosal tissue and apply the API solution, or at the end, the oral thin film on the donor site. So we have here the 6-well plate as acceptor compartment and the insert with the mounted mucosal tissue as our donor compartment. We have very short sampling time points because they only need seconds, because we can dip one insert from one plate to another plate. And what else is an advantage that we have with a specific shaker, the possibility to heat up the system to body temperature, and we have the possibility to move the whole system. So this is a little bit different to what we normally have if you apply a patch. So if you apply a patch to the skin, then more or less this application, let's say, area is static. But in the oral cavity, we always have the dynamic movement of the jaw muscles and also the tongue. And so this has a main influence on the solubility of the API from the oral thin film. And it has also an influence on the permeation rate we expect for this API. And so this is good to mimic at all. I mentioned before that you also have the possibility to apply something sublingual, and that you might have more spreading for your API there. And so for this, we need another model. And we designed a model which we call the drum, because this is exactly what we have here. So we have our inserts again, and we mount or apply the mucosal tissue of both sides. And then we have the donor compartment in the middle where we can apply our API solution, and we have the acceptor compartment outside. So we have here the possibility to mimic the whole oral cavity. That's also interesting if you have an API with a very high speed of permeation, for example, where you think about that you make a fast dissolving film at the end, where you have a distribution in the whole oral cavity and not only on your buccal side, for example. And you can investigate a spreading of an API could advance your permeation. Also here, we have the same procedure like we saw before. We are normally a little bit faster than for the feasibility studies of the Transdermal therapeutic system candidates, because we don't have any possibility to mimic something like PAMPA. So we investigate a lot of different ideas, but there at the end is no artificial membrane which simulates very good the mucosal tissue at the end. And so this step is not placed in this feasibility study. We have to focus in the feasibility study itself, and I will come to this a little bit later in the examples, on some other points. So one point or one main topic is the solubility in saliva and the influence of saliva, because in the oral cavity we have saliva at all and all the time, and so we must investigate this too. We also need HPLC methods at the end, and of course, we also have the possibility to screen enhancers, but we don't have so much enhancer in our portfolio than for the Transdermal therapeutic system, and there are also only a couple of enhancers described for oral thin films, and which work very well. And so at the end, we are normally a little bit faster than for the Transdermal therapeutic systems. I mentioned before that we use mucosal tissue from pigs And we don't have the possibility to use mucosal tissue from Göttingen and Mini because the tissue region is too small for our experimental trials. So we use the mucosal tissue from domestic pigs, and it's the same like we saw for the human skin. We have a very high variability from donor to donor. What we normally do is that we mix up different donors, and we increase the number of replicates, and we set a reference. In this case, normally it's very easy to set a reference because we normally use a dedicated API concentration, which is soluted in saliva or in artificial saliva. If we have questions which derives to a higher discrimination potential of our model, then we normally go to the reconstituted cell cultures because they have all the same genetic background, and our standard deviations are much lower than using the mucosal tissue at all. What we have to investigate in case of the feasibility study of an API for oral thin film is the dependency on pH. So we normally use the salts of this API, and it can be that we have an interaction of the pKa values of the API and the pH we normally want to use. And so it's really necessary to investigate this in the beginning because depending on the diet, normally customers or patients prefer the pH value in the saliva can change. And so it's necessary to investigate what happens in a neutral and in a slightly acidic condition. And if we saw a difference like we have in this example, it might be a good idea to, at the end, to make a buffer system in the oral thin film, for example. So it's good to know for the development department if we have a pH dependency or not. I mentioned before that there are only a couple of enhancers. This is a little bit based that we don't have so lipophilic substances, and most of the enhancers are very lipophilic. And so we have sometimes a problem that they might be a good permeation enhancer, but they have no or no less solubility for the API. And then you have the problem that you cannot combine a very lipophilic enhancer with an aqueous system like we have in the polymers we use for oral thin films or something like that. So it's necessary to set a reference and then look if we have the possibility to increase the permeation potential of your API in combination with an enhancer. But we all, as mentioned before, we only have a couple of enhancers which we can investigate here. So this is a field which is more new, let's say. Sometimes if we have lipophilic properties in our API, not so lipophilic like we have for the Transdermal therapeutic system, but it could be, then we normally saw in our models that our permeation is very slow. And sometimes you also ended in something where you don't have any differences in the permeation at the end. But then it might be a good idea to look in the reservoir or in the depot of your epithelium tissue, because in case of these lipophilic properties of API, the application time of your API or at the end of your oral thin film must not be the same time that you have for the permeation. So if you have an increase in your mucosal tissue, then normally permeation of your API takes longer than the application time at all. And so we can develop, or we can investigate this by the investigation of your API concentration in the epithelium tissue in different sampling time points. And you can see here that we have a good increase when we use this special enhancer in this case. Sometimes it's necessary to make an individual adjustment of the in vitro permeation model. So for example, if we have to handle a very big API, so big molecule size, then sometimes it might be a good idea to look if there is any benefit using let's say a spreading of your API. So at the end, for example, a sublingual application. And like you can see here in this example, we permeated in our normal 6-well design, and then we go to our 3-day model. There we only have a double of the diffusion area, but in this special example, we have an increase of permeability of factor of 10. So it might be a good idea for this API to go at the end to a very fast dissolving film, which can be applied under the tongue to have more spreading conditions for this API. We have to discuss also enzymatic activity. Normally, the APIs for transmucosal applications have a very high potential. And so we have to investigate if we have any influence by enzymes. Do we have, for example, an active transport which can increase the permeability, or do we have something which metabolizes our API very rapidly, or do we have an inflammatory, let's say, inflammatory effect where macrophages will transport the API to another region and not to the vesicular system. And this is the case in this example. So we investigated the API solution on our 4 constituted epithelium model. And we saw that the active version of this model might influence the permeability of the substance in comparison to the inactive variation of this model. And this normally is an inflammatory process or something which has to do with the macrophages. And then we have a transport of the API. So if there is a hint that your API has a high potential or is directly involved into metabolism process, then it's a good idea to also investigate this in a very early stage of development, so in these feasibility studies. At the end, I want to show as last example, an example where we have the indication for an active transport of the API, which increases the permeability normally. And we can simulate this by using the reconstituted epithelium here in green. But we also have the possibility, if we know the enzyme for this active transport, to add this enzyme to our mucosal tissue model and see the same effect. So this is normally what we do in the feasibility study for candidates for oral thin films. And to sum up this part, we normally evaluate the solubility of the API, the pH dependency, the solubility in saliva, and the permeation potential. Sometimes improves by enhancer, but the number of enhancers for oral thin films is much more less than for transdermal therapeutic systems. With the variability in our models, we have the possibility to use individual conditions for the API, and we also can investigate which potential application site at the end or spreading might have a good effect on your API penetration. Now, after a lot of information, I think we come to the conclusion at all. René showed you in the first part the tool PRAISE, which has a possibility to evaluate the suitability for a drug candidate for transdermal or mucosal route. I showed you the practical approach for both candidates for transdermal therapeutic system and also for oral thin film, and what we normally do within 3 months. So we have a very fast evaluation process here, and at the end we normally have also the possibility to have a go or no-go decision for further formulation development. And with this combination of tools, we have the possibility that we only need less amounts of API. This strategy is innovative because we only work with models and techniques which are really state of the art in the moment. And with all this knowledge let's say investigation we make about the suitability of an API, we understand very well what the characteristics of the API are and if they have the potential to be a transdermal or transmucosal drug at the end. So we are finished with the webinar today. Thank you for the audience for listening, and we are now happy to answer questions.
And thank you very much for this presentation. Just a reminder for the audience to ask questions. You can send them via the questions widget. Just type them in this box and click submit. Please note that you can still send your questions after the webinar and they will be answered at a later date. But it looks like we've got several questions already, and here's our first one. What happens if only limited data for a trace evaluation are available?
So thank you much for this question. Let me jump to the trace analysis for azanapine. Let me check. Can I jump now back? No. Okay. You can see here we try to include many different specific information to one trace analysis. So if only limited data are available, for example, for a new chemical entity, you usually know the molecular weight, you maybe have a rough idea on the dosage, but no information on the oral bioavailability, we could perform such a trace analysis, but for sure the significance would be reduced. If the available data are still valid to perform an transmucosal or transdermal feasibility study, for example, the molecular weight is not above 1,000 gram per mole or something like this, then we would recommend to initiate a short feasibility study, as Julia explained you in detail, and see what we can see really in vitro for your transdermal and transmucosal candidate.
Thank you very much for that response. We have another question from the audience. What is the preferred receptor composition?
Okay, I think it's a question for me. So it's a little bit depending on let's say, which API candidate we are talking about. So in case of a candidate for transdermal therapeutic system, we normally choose an aqueous buffer with a pH value of 5.5, because this is the pH value of the skin surface. If we have problems with the solubility, it's also feasible to use a pH value which is physiologically, so 7.4. pH values under 5.5 and over 7.4 are normally not the best way to choose for the receptor compartment. For the oral thin films, we normally work with a receptor which has the pH value between 7 and 7.4, because in the oral cavity we have more or less the physiological conditions.
Thank you very much. And here's our next question. How much API is needed for such feasibility studies?
Maybe I can answer if you don't want, René. It's a little bit dependent on how good your API perform. But let's say overall, we can make the first feasibility trials overall with, let's say, 10 to 20 grams of your API. And then we have many opportunities to make this feasibility study.
Thank you very much. And we have another question from the audience. Okay, what quality parameter for the evaluation of skin do you use?
Okay, quality parameter for the skin. Okay, if we use skin, and I mentioned before, we have a high variability from donor to donor. It can be that we don't know this from the plastic surgery, for example, and that we don't have the same dermatological quality for the skin at all. Regarding the guidelines we normally have, we have the possibility to measure the transepidermal water loss as one parameter, big limits from dermatological experts, which gives us a ranking. So the transepidermal water loss is an indirect proportionality to, let's say, the thickness of stratum corneum, or you also have the possibility to see any leakages in your preparated skin, for example. We also make something which we call internal permeation. So we make an internal permeation experimental trial where we have something like a ranking for the permeability of each skin donor we get at the end. And so we have the possibility, for example, if we want to see which our value from permeability potential for an API, to choose a skin with a very low permeability potential and a skin with a very high permeability potential at the end.
Thank you very much for that response. And we have another question from the audience. Maximum, how long can we run IVPT without compromising the skin barrier?
Yeah, I think it's a question for me again. In case of in vitro permeation studies for Transdermal, therapeutic system or API candidates, we have the possibility to work with the skin up to 7 days. Without any influence on the quality, if we design the frame conditions as good as we can. So there might be an influence if we have to use a high concentration of solubilizer in the receptor, where we have an influence on the quality of the skin. Then it might be the possibility that we only can apply or use the skin for 3 days. In case of the mucosal tissue, we don't want to use them longer for one experimental trial than normally for 8 hours. We saw there is a really big influence for overnight permeations, and we saw that the barrier function is not as good as we make it when we use it in a fresh way, let's say.
Thank you very much. We have time for one more question today, and our next question is, for the PAMPA pampa, what kind of membrane it uses? Is it cultured cells or just artificial membrane?
Okay. It's dedicated by the supplier of the pampa system, but it's not a cell culture. So it is an artificial membrane, and they have a special printing, let's say, doing where they can apply the main lipids of the stratum corneum and include this into the membrane. And we also investigated the lipid composition of this membrane in comparison to the lipid composition of stratum corneum, and we saw that we have the main or the same main lipids, and also the quantification of these lipids is very similar. So it is a good tool to mimic the stratum corneum as barrier, but it don't have the possibility to mimic the whole epidermal layer, so you are not able to see any depot effects or other influences you can have from the skin.
Okay, we have a few more minutes before the end of the webinar, so let's check if we have another question today. What is your choice, like only stratum corneum or full skin?
Okay. We normally don't work only with stratum corneum, or let's say it's not easy to isolate only stratum corneum. And we have the problem that it might be then very thin and that we have many leakage problems. So we have to increase number of replicates and so on. What we can do is we can make a heat separation to only work with the epidermal layer, but we normally work with a dedicated skin thickness, and it's a little bit depending on what you want to see. We can vary this from 400 to 800 micrometers normally.
Thank you very much. And just a reminder that you can still send any questions while watching on demand. All questions will be answered at a later date. I would like to thank Julia and René for this presentation and LTS for sponsoring this session. To the attendees, you will receive an email telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is pmi-live.com. We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. Thank you once again, and I hope you all have a nice day.